Hi, everybody. Thank you for joining us this Friday morning for our stages trainees presentations on the work that they've done throughout the summer on their projects in the Big Data Blitz. So over the course of the summer, the trainees have taken their projects and worked on acquiring the data sets for them and then performing quality control and assessment of those data sets, looking at the temporal and spatial differences and trying to get everything aligned so that they can move forward this fall with their AI and machine learning models in the environmental forecasting class. Our trainees over the course of the summer have also completed multiple micro-credentials through the Texas A&M's High Performance Research Computing Center. Those credentials have been Python-based predominantly in various becoming more familiar and competent in Python. And then there have been some more advanced ones in AI machine learning, deep learning, using TensorFlow and other options. So with that, I would like to invite our first group up. We call them our bacteria group. This is Laura, Ifani, and Quincy.
I'm going to start it, Bonnie. We're ready.
Right. Hello, everyone. My name is Ifani Imalanyang. I am a first year PhD student in coastal and maritime science. And my research interest is focused on understanding the impact of flood events on water pollution in unincorporated Gulf Coast communities. I will give opportunity to my colleagues to introduce themselves.
My name is Quincy, and I am a master's student in the CMSS program, and my research lies in using radar data to understand the properties of extreme precipitation events.
And my name is Laura Button. I'm a second-year master's student, and my research involves groundwater contamination with unincorporated coastal communities.
All right. So the topic of our project is identifying key drivers of Enterococci in Texas coastal waters. So Enterococci is actually a gram-positive fecal indicator bacteria that is found in water bodies, and it's actually sourced or could be linked to the presence of septic systems, wastewater influence, and other anthropogenic sources. So these bacteria have been discovered to not be harmful, but they are key indicators of fecal contamination in the water bodies. The Texas Beach Watch program is actually a body that is vested with the responsibility of monitoring the beaches along the Texas coast. And so some studies have been done recently that have been able to identify some dynamics in terms of the bacterial loading in the Texas beaches. Powers et al. 2020, Powers et al. 2021, and Kai et al. Have been able to identify that the fecal bacterial loading in these beaches is highly influenced by sea level rise, population size, stormwater runoff, and time as well. So the key aim or the key goal of our project is to use AI and machine learning to model distinctive bacterial loading patterns and identify some bacterial peaks that are really not easily identifiable along Texas coastline, looking at the sets of data sets we have to work with, and also link it to the influence of environmental drivers and other factors that would actually influence these dynamics. Next slide. So the study area for our project is actually 4 counties. The last meeting we had with TGL, we were able to streamline down to 4 counties: the Madagora County, the Brazora County, the Galveston County, and the Harris County. So these counties have been identified to have high Enterococcus concentrations, and as such, it's very important for us to look at these areas and also try to identify key drivers that are influencing these high bacterial peaks in these counties. I would hand over to my colleague Quincy, who will take us over with the sources of data that we have. Thank you.
All right, so I'll be talking about the data sets that we have and how we plan on implementing them into our AI model. So starting off with our primary data set, we have the bacteria data from Texas Beach Watch. And from this data set, we're able to obtain the Enterococcus measurements as well as where and when they're recording these samples. And this data set spans from 2009 to 2023. And then the next data set that we have is a cell phone density data. And so what this data set does is that it essentially tells us how many cell phones are within a given area at a given day. And one reason why we're using this data set is because our community partner is very interested in seeing the linkage between people and bacteria levels in the water. And so this data set will give us hopefully a better insight into this linkage. And this data set spans from 2022 and 2023. And so for at least this phase of our analysis, we'll just be looking at these 2 years so that we can give a fair comparison between bacteria levels, people, and as well as precipitation, which I'll be talking about next. So the next data set that we have is the MRMS precipitation features data. So this is all the radar data attached to individual rain events that were detected by ground-based radars. And so what we obtained from this data set is, of course, the location of each rain event, as well as some of its radar-defined characteristics, such as the rain rate, the area of rain coverage, and other characteristics such as the volume of rain dumped over the area, as well as how much liquid is within the storm itself. Then the next data set we have is the septic system data. So we're able to obtain the locations of reported septic systems within the Texas coastline. And then we have our tidal and meteorology data from NOAA. So we get the water levels as well as wind speed and direction. And so what we could possibly look at is we know what direction the wind was coming from the day that a sample was recorded, and then they can possibly see like what sources were coming from the direction of the wind. Then we have the potential point sources of contamination identified by TCEQ. And so from this data set, we have locations of these identified sources as well as a description of each source. So these descriptions can range from agriculture, such as a farm or like a well-known fishing spot, to something industrial, such as a wastewater pipeline or wastewater treatment facility. So this is just a visualization of how much rain data we have to work with. So another aspect that our community partner was interested in was identifying the linkage between rainfall and bacteria levels. And so this is just a visualization of all of our rain events that we have to work with during our years of study. And each individual spec you see in this figure represents an individual rain event, each with their own set of unique characteristics and values. And so all in all, we have a little over 2.9 million rain events to consider for our analysis. And then because we have so much rainfall data, we're able to take a closer look at identifying this linkage between rainfall and bacteria levels. And so this map or this plot just shows a comparison, like a rough comparison between rain events and the bacteria levels. So each of the bars represents a count of rain events detected within our areas of study. And then each of the colors represents a different rain rate category that they fall in. And then the red line overlaid is just the mean bacteria level for the month overlaid over the top. And so what we hope our AI model can aim to answer, or at least give us some better insight, is, for example, like in the first half of the year or the first few months of the year between January through May, you can see that we have relatively lower counts of rain events, but you see a spike in bacteria in March, and then as well as one in May. And so we know in March, that's when spring break occurs for the state of Texas. And so a lot of people are flocking to the beaches. And so is this spike correlated with people more than rainfall? And then same in May, this coincides with summer break for a lot of schools and people in the state of Texas. And so is this spike in bacteria related once again to people more than rainfall, or is it related to a different source of contamination? And then looking at the later into the summer and in the fall months, you can kind of see that there might be some kind of correlation between rainfall and bacteria amounts. So you can see a spike in September for both rain and bacteria counts. Then you see a dip in October and then an increase in both in November and so on. So we hope our AI model can help us explain what kind of patterns that we can or cannot see between all these different variables. So this plot is just like a visualization of what we would put into the model essentially. And so we have our test site given by the black ring in the center of the figure. And this was a test site in Matagorda, specifically at Jetty Park number 1. And then the sample was recorded on July 1st, 2020. And so what we did was we pulled together all of our other data sources and then try to find what sources of contamination were within the test site within 5 miles and within 7 days prior of the sample being recorded. And so what we have identified was a few rain events given by the multicolored circles, and the colors of the circles correspond to the rain rates in the color bar. And then we have the septic systems identified within a 5-mile radius of the test site. And then from the TCEQ point sources of contamination, we have identified 2 sources of contamination, one being a source of wastewater and the other being a wastewater outfall. And so when we put these variables into the model, one thing we want to look at is how many features occur within a certain mile radius of the test site, as well as the proximity of each feature to the test site. So I guess how close the septic systems are to the test site or how close the rain event is to the test site. So I'll pass the mic over to Laura and she'll talk more about our cell phone data.
Thank you, Quincy. So in terms of the cell phone data, this is just an example of what one day looks like in terms of the density and our study area. So as you can see, May 3rd, 2022 is Tuesday. That's kind of an odd day for the top usage in the whole summer months of Texas. But most of it is condensed in this Harris County area, right up here. And something that we found interesting was in terms of the top 20 days of cell phone density in 2022, a majority of the days are in May and April. And with that being said, that skips out June and July, which are some of the top beach months. That skips out Memorial Day, 4th of July, and it puts it all mostly in Clear Lake. If you go back to the map, and Clear Lake, it's just about this area here, which in Texas Beach Watch, that's not necessarily tested for. So that's already giving us insight on where we can kind of plug in information, take kind of estimates on that, but also in terms of future testing for Texas Beach Watch, that in my opinion is a pretty high area, considering that in one of the graphs that Quincy showed, May had a giant spike without any rain for the most part. So anthropogenic causes could definitely fill in some gaps with that. But overall, for our workflow, we plan on, or as Ifani mentioned in the beginning, our objective is to identify the key drivers for enterococcus levels. And as Quincy mentioned, for all of our input variables, we have many data sets to work with, and to keep adding with each other into the model is going to give us a lot to just look at overall. And then for our methods. So right now we're in the first 2 bullet points. We're still cleaning up the data. We just got the rest of our bacteria data, so that's very exciting. All we have to do is clean it up a little more and put it into a CSV. And then currently we're isolating the storms and the beach events. So that's something that I'm personally going to do with the top 20 days, is to see if any of those are correlated with events in Clear Lake, and to see if any of that makes sense, or if they're just random high density beach days. We'll use that to train the model, as like basically we know these days are caused by, for the most part, people or storms. Then we'll create the model and then keep training it and perfecting it as much as we can by December. But overall, our key findings are to find those key drivers to see, what is causing these beach watch advisories, and not only, you know, define them, but to also try to keep the public as safe as possible with knowing like if there's high rain or if it's a certain time of the year in Clear Lake, then we know that we should probably keep an eye on that area to keep, you know, beach watch advisories up, or to know where to put the attention at essentially. But okay. So we'd like to acknowledge, you know, obviously our wonderful partner, TGLO. We've been able to keep up great communication with them, SAGES program and the NSF. But these are our references, and thank y'all for listening. Y'all have any questions?
So,
Hello?
wait, some online.
Hey, y'all, this is Jason Pinchback, Texas General Land Office. Thanks for sharing the information. It was really insightful, and we do appreciate your work. I really look forward to the results as y'all keep on to build your knowledge base. And it's going to be really interesting to see how your more sophisticated way of capturing rainfall data is going to inform us. And I couldn't help but to wonder, you know, what some of your data your rainfall data would compare to, like, for instance, I guess it's going to be basically rudimentary, but accessing the text mesonet data. And just it's more of a curiosity on my end, is like, how much information are we missing by just relying on the text mesonet for precipitation data? So as y'all kind of cook these stats out, I'll definitely be interested in kind of drilling back in time and doing some queries out of that, just to kind of see how things look. But great work. I appreciate what you're doing, and really learned a lot this morning. Thank you.
Thank you.
Dr. Liu?
All right. I thought you would answer the question about the difference between the Mesonet and the MRMS. Jason was asking.
Yeah, so the mesonet just records like the ground base of rainfall measurements. So that's how much rain was accumulated on the ground. And then the MRMS is just the radar data. So what's going on above.
So my question is about when you build the AI model and everything is rely on how much data you get and what's the temporal and the spatial resolution. And of course, your target is sporadic, those ground stations, and you have no way to change that. But what is the temporal resolution of your bacterial data, and how would you corresponding that to your cell phone data and also the MRMS data, because those are in different temporal resolutions, and many of those courses and results maybe have a time lag in between, and those are the challenges. And I just want to know how you think about it, what your thoughts on and the plan for addressing those.
Yeah, so right now we're still in the phase of identifying these temporal overlaps between all of our data sets. But we do know that the MRMS data set takes a sample every 6 minutes. And then the Texas Beach Watch, they record a sample, I believe, once a week at each site.
2 weeks, I think, yeah.
And then the cell phone data, we have a density value given for every day between the 2 years. And so we're still trying to identify what kind of overlap we want to do and how we want to correlate all the events together for our 2 years of study.
For the bacteria data that's being collected, do they report error percent or rates with that? And if not, is there an average understanding of like how accurate are those measurements to the actual value?
They don't record an error percentage, but at least with the Mergulet lab, like we've worked with the enterococci data before and everything. So that's not gonna be difficult to get that error margin. Many of the students in the research lab have used that before. So there's a general understanding, but thank you for bringing that up. That's definitely something for us to add in there. Alrighty, well, if that is all the questions, we appreciate the feedback.
Thank y'all.
Dr. Tisto actually had a question in the chat. Is the MuseNet data somehow included in the calibration of the MRMS model? I.e., is the MRMS somewhat informed by the MuseNet data?
Lindsay, you want to answer or I should answer?
Yes, if I'm not mistaken, the MRMS data set is kind of validated by the Texas MesoNet data already, so.
Yeah, it's inside. And
Okay.
so they use the MesoNet data ring gauge to do the calibration, but it's calibrated every hour.
Yeah.
So it's, even though it's a 6-minute data, they put hourly ring gauge data to use the total to adjust it. And yes, the answer is yes, but yeah, MRMS has much higher resolution though.
Our next group is the manta ray group, that includes Mahima, Amanda, and Remy.
Morning, everyone. Thank you so much for being here. As Dr. Douglas introduced us, we are the monitoring race for improved NOAA enforcement. We're the marine group, and this is our final presentation comprising everything that we completed this summer during the Big Data Blitz class. I'm Mahima, this is Mandy, and this is Remy. We'll go ahead and start by introducing our research backgrounds, and then we'll get into the meat of what it is we're doing. So as I said, my name is Mahima. I am a master's student, a second-year master's student pursuing a degree in marine biology, and my interests are marine microbiology, diazotroph ecology, and molecular biology. I'm going to go ahead and hand it off to Mandy.
My name is Mandy Burton. I'm getting a master's in chemistry. I focus on seagrass genetic diversity, alkalinity, and groundwater and estuarine systems. And I'll give it to Remy.
Hello, my name is Remy Albay. I am a master's student also in the chemistry program here at TAMU-CC. My research focuses on epigenetics, multi-omics approaches towards understanding how toxicants affect marine organisms as well as human organisms. And I'll pass it on to Mahima.
So before we get into the data analysis portion of our project, we wanted to talk about the study species, which is Mobula barastris, also known as the giant manta ray. If anybody's interested in the taxonomic classification, we won't get into that, but that's listed all the way on the left here. One thing that I did want to point everyone's attention to that Mandy will mention later on is that one of the most unique parts of the manta rays are these spots on their underbelly, which are unique to each and every organism, and we'll circle back to why that's significant. Go to the next. So just some basic stats about the giant manta ray. It has a wingspan of up to 26 feet and can weigh up to 5,300 pounds, really living up to its name of a giant manta ray. These are migratory fish, though there are some knowledge gaps that exist on maybe where they migrate and where a majority of the population might lie. They do have a lifespan of 45 years, but they do have a low fecundity, meaning that they only give birth to pups once every 2 to 3 years. But the most important thing here is that they are classified as threatened under the Endangered Species Act, necessitating that we take further measures to understand how to protect them. Their main threat is commercial fishing, where they're kind of caught as bycatch during these shrimp trawling nets that catch them. And specifically, sometimes they are specifically harvested for their gill plates. Next slide. And this is a map that we pulled from the NOAA website, kind of outlining the main distribution of the manta rays. So they are kind of seen just about everywhere, but again, there are significant knowledge gaps existing on maybe what their migratory patterns are, what their development might be, and so that's what we're aiming to elucidate here. So I'll go ahead and hand it off to Mehdi. Or Remy, I'm so sorry.
All right, so our plan is very similar to the little game or the game that everybody's familiar with called Where's Waldo? But in this case, we're going to be playing a little different game called Where's the Manta Ray? And throughout these slides, there's going to be mantas. And everybody, if you guys get bored, just play this game called Where's the Manta Ray? But in all seriousness, we're going to be doing the exact same thing as like the game I said, Where's the Manta? But instead of having the human's eyes do this identification, we're going to train a computer to do exactly just this. And the way we're going to be training it is through a platform called YOLO V8. And the input imagery data that we're going to be using is satellite imagery. So, and I'll pass it on to Manny.
So the reason we're doing this is to track the movement patterns of manta rays in general. But there's also a fun little thing we might be able to do where we could possibly identify the movements of individual mantas with those underbelly spots like that we mentioned. Because apparently mantas like to breach and flop on their back. So if we see a bunch of pictures of the same manta ray on their back, we can track where it's been. But we're mostly just doing this to track the species in general. To confirm or reject the location predictions that NOAA made. The problem with these is these are sightings data. This is a compilation of sightings, and it's hard to know if it's just they're there more often or they're just people there more often to see them. So with the satellite images, we can kind of venture farther out from the coast and see where they actually are and how often they are there. We hope to do this to reduce bycatch, because if there's a manta that's always in the same area, we can probably tell them like, hey, don't trawl over there today. This is the season that they're there and they are always there. And I'll pass it on to Brendan.
All right, so the workflow that we're going to be employing here is one that's very simple. We're going to be taking a satellite imagery data set and then annotating that data set to have images with a box around the manta rays, at least 100 images ideally. And with this image data set, then we put it into the YOLO V8 training platform, which runs on PyTorch and a number of other packages like Ultralytics. And through the training, basically what we hope to obtain is a refined AI model that's capable of identifying these manta rays for this specific type of satellite imagery. So we're going to have to stick to a specific image size, length and height wise, and then identify the manta ray as our output. And the workflow here that I have is very simplified, but once we have this trained AI model, the goal would be also to develop a software or a very simple algorithm that can be used in the command line, in which our stakeholder NOAA can very easily put in arguments of where their images are in, what library and what's the path, and then perform that analysis, define an output path, and then have labels as well as data about where locationally, where was where were those manta rays detected and when, based on the image annotation. A little bit more in depth as to how we do this AI training model. We take, like I said, a training data set, in which is very simple. It's just an image, the image data set containing the mantas, and we have a little box coordinate as a separate file, which determines exactly where that object, the manta ray, is. Once we have that data set, what we do is very simple, one line, say YOLO V8, we want to train on this image data set, and then we train the data set, and then after that's been done, we have to validate. This is a very important step and usually requires a lot of refinement as to what our parameters are for the training. So if we don't hit our threshold of 95 up to 98% accuracy or confidence that our AI model is able to identify manta rays, then we repeat step 2 of the training to refine that model. And then once we have accomplished that 95% confidence in our AI model, then we will deploy it to that program for our stakeholders to utilize. And now I will pass it on to Mahima.
For me, so we wanted to talk about some roadblocks that we've encountered so far during the summer. So in terms of what went wrong, I wouldn't say this was so much as what went wrong as something that presented slight difficulties. So our stakeholders did try to give us as much data as they could, but unfortunately it was not the exact Maxar satellite imagery that we needed. And there was a bit of a tie-up and kind of a delay in getting those images. We do finally have them now, and we really do appreciate our stakeholders for working so incredibly hard to get us those images. But because we didn't have them from the get-go, that required us to be flexible and try to figure out different kinds of pivots in case we did not have those pieces of satellite data, so that we could still complete the objective of the project. So in terms of what went right, though, we were definitely great at communicating, setting reasonable goals, and being flexible in the midst of these challenges. So I'll go ahead and pass it on to Mandy, and she'll talk about some of the ways we pivoted.
Okay, so we had a couple contingency plans. The first one we had was to use Google Earth Engine images because they're freely available and there's a command line in there and you can put your code right in there. But ended up the resolution was not nearly good enough. It was about 10 meters per pixel and a manta ray is max 8 meters. So it'd be less than a pixel, and then to prove that it's a manta ray and not just a shadow or anything else is kind of impossible. That'd be a bit of a stretch. So we decided maybe to try using aerial images that NOAA had from an aerial survey where they purposely took pictures with manta rays and scaling the resolution down to what a satellite would look like and changing like the color gradient to make it like a mock satellite image to train the AI for that. And then the stakeholders who have access to the Maxar could deploy it on there. And we were in the beginning stages of doing this when we got the Maxar images, so it ended up not being necessary. But we are going to keep it under our hat just in case we can't find the 100 necessary threshold training images, so we can do that to kind of supplement and maybe even add a little variety to keep the AI sharp. So the Google Earth image approach, what I did was I wrote a couple codes. We were given sightings data from a paper written by Nick Farmer, which was that map I showed you earlier of all the sightings, when and where they were. Unfortunately, they did not match up time-wise to when Google Earth Image took the pictures. So I did some statistics on the sightings Excel to find out what months were most likely and where to narrow down the plethora of images for Google. And then I tried to run a match with all the images. I made a list of Google Earth images, but first I had to isolate them so they weren't all overlapped. Isolated them and then put them on the Florida coast and then made sure they had the ocean, because Florida's at a diagonal. So it might be ocean here is completely land here. So I had to do that, which was honestly the most tedious part, but I got it done. But like I said, the Google Earth images weren't quite sharp enough, but the code's still pretty useful. And I made a GitHub for anyone who wants to do anything remotely similar. This is what the Excel sheet looks like when Google Earth Image spits it out. So you get the image name and it's got the all the numbers they put the name on, and it's got a date. And then it'll give you the linear ring of the outline of the picture. And so then I ask it to find a picture that's in that circle. Now I'll give it to Rin.
All right, so now that we have the Maxar imagery data set, we will try to port through the gigabytes of photo imagery to find these manta rays. Now, this is going to consist of hundreds, if not thousands of images that we're going to have to port through. And to refine that search, we're going to use a little tricks like the water anomaly detector, which is another AI model that was previously developed that is capable of identifying water anomalies from imagery of the ocean. And so we're going to try to first use that to find anomalies such as the manta ray to narrow down our search. If that doesn't work, then there are other things that we can do, such as manipulating the color thresholds to identify dark spots in the imagery set, such as a giant manta ray, and be able to basically identify manta rays using a threshold, and then we'll visually validate that for then the training data set in order to obtain our 100 images. Now, if we fall short of 100 images, what we can do in the YOLO V8 pipeline is rotate the images. So what images we have, there is a short command for how many times do we want to randomly rotate a confirmed image for training. So this is a way to kind of artificially increase the our training data set that we will do. So yeah, so that's pretty much where we're at and where we're moving as of I think August, right? So yeah, that's what we're going to be doing. And then I will be passing it on to Makima.
Yes, so this is kind of a summary slide talking about everything we've already discussed. So we want to make sure that we have everything that we can possibly have right before the start of the semester. As we all know, it gets incredibly chaotic. So a couple things that we want to do are maybe parse through at least some of these Maxar images that we've gotten, just to get an idea of what we're looking at. We know that it will be extremely time consuming, and so we want to set realistic goals for ourselves. But now that we have the images, we're hoping to potentially meet with NOAA more frequently, which is something that we will email them about, because we want to just make sure that we're hitting all the goals. And we'll continue to prioritize being proactive as we have been, and doing everything else that we have in order to have a fully fleshed out presentation and have it be as robust and informative as possible come November when we do the final presentation. On to the last part. We wanted to go ahead and thank Stages and TAMU-CC Christi for offering us the opportunity to be in this fellowship, and our wonderful collaborators at NOAA Southeastern Fisheries for presenting us with this really exciting project, and we're extremely excited to see what comes of it. And with that, we'll go ahead and open up the floor for questions. Thank you.
Thank you.
Yes, Dr. Lowry.
Let me stand up so I can be seen and heard here. I was interested in sort of looking ahead. I was interested in what you intended to do at your validation stage. How do you intend to validate your model?
There's an internal command in which you take the train model and it runs through either the same training data set if you don't have enough images, but ideally you would want excess images that you have valid manta rays in, and then you would have the AI go through those images, and you have, of course, the answer of the box, and if it's within that box coordinate, then a percentage is given. As to exactly how the algorithm does that, I'm not quite sure what that's about.
Well, something you might consider, this is sort of pie in the sky. Since you're running the anomaly detector anyway, is to save some of those images as well, because what you don't want to do is identify fishing boats as manta rays. And so those would be good things to run through as a validation step also.
That's a good suggestion. We were also going to, for the validation step, you also give blanks in which you don't have the, like you said, false positives, but also blank images.
Of course. Yeah.
Right. Okay, thank you very much. Yes.
Is regarding the 100 images, is that like a hard limit? So assuming that the detection algorithm works to like significantly cut down on time of manually looking through each image, like could you incorporate even more than 100 images?
Absolutely. Ideally, you would want more than 100 images. 100 is kind of the given limit, minimum or training an AI model. From my experience, depends on also the object itself. How easy is it for the AI to identify the object? If it's really easy, then the less number of images you actually need. The harder, the more images you need.
But ideally more would be great, because even when we were going through the Google Earth images, it was so easy to mistake something else for a manta ray, which is why we quickly realized that going through it with, human eye is a very difficult thing to do, and presents its own set of unique challenges. So
Yes, Dr.
So what is the resolution you got, the data, Maxar, the spatial and the temporal?
I believe it's 30 centimeters per pixel. That's what I believe. So if not, it might be the 1 meter per pixel. It depends on what plan they chose.
How about the temporal? Do you have a
Oh,
same location scan second time?
so they have a whole archive of all their images, and we sent them a latitude and longitude range we'd like the images from, and the months that we want it from. But we haven't gotten a chance to look at them yet because I'm working on getting ArcGIS on a computer here. But they've already been kind of refined by John Quinlan. Thanks, John. That way.
One suggestion is that because Monterey is moved, so if you compare the two images on the same location, if you find the differences, and then those hotspots will stand out. Of course, that doesn't separate the boat and the Monterey, but at least you know that it's something you can use your eye to look at. So you can pick those out and use that's a easier way to pick out your target instead of staring at it all the time.
I just have a suggestion for you guys. If you want to use YOLO V8 model, you have different techniques of data augmentation, as you mentioned before. You want to increase your images. Try to just use that different techniques like flipping or rotation, or also mosaicing, data augmentation. This is so important. If you want to have more images, just try to use all the techniques you have for that model.
You
Yeah.
definitely do that. We want it to be kind of how Remy said, keep the model on its feet and, or I guess on its toes, and just try to manipulate the images as much as possible to increase its ability to detect.
One thing I did not mention, but there's also different sizes for the YOLO V8. So you have nano, regular, and then mega. And we'll be ideally training it on the HPC at the highest level. So the largest algorithm that we can run.
Yeah, and you also can just mosaic 4 images together and just do the model and train it again. And yeah. Yeah.
Do we have any questions from any of our online participants or any stakeholders? I'm gonna open up the chat just in case. Stop sharing. Are we?
You don't need to stop sharing, but if you go to the top, it should drop down. The top middle.
Over that green, yeah.
Oh, worldview one. revisit.
Oh, right.
Yes, yes. Yes, thank you, Dr. Quinlan. But yeah, if that's, that's all, then we'll go ahead and, stop talking. Sorry, we don't know how to transition out of that. Thank you all.
I'm gonna go.
That'd be cool. You could track it.
Grace and years.
Okay, our next group is our shorebird group. That includes Allison and Justin, but Justin is with us in spirit today, but not in person, because he's up in the middle of Lake Erie, I believe. Doing field work.
All right, good morning, everyone. My name is Allison Gerard. I'm a PhD student here at TAMU-CC in the Coastal and Marine System Sciences program. And I'm joined by my group member, not in person, but Justin Elliott, who's been working on this project with me as well. And we've been working with Coastal Bend Bays and Estuaries as our primary community partner on our project titled Identifying Inland Wetland Habitat in Rural Southeast Texas for Migratory Shorebirds Along the Central Flyway. So Coastal Bend Bays and Estuaries came to us earlier in the spring and expressed to us this long-term goal that they have of developing some sort of incentive program such that we can motivate private landowners to manage their land in a certain way that is amenable to supporting migratory bird foraging. And we're sort of drawing inspiration from a similar program that already exists out on the West Coast called Bird Returns. And this Bird Returns group has several different incentive programs to offer maybe financial incentives or protections to private landowners who can work with them to manage their land in a certain way. So these programs that are already in place with the bird returns group are built on a strong foundation of research and analytics to inform this group of how they can reach out to the landowners, who they should reach out to, and the different strategies that they could use to manage wetlands in this way, and what sort of incentives would be useful for that objective. So there's a lot of research that's already gone into this by this group, and there's this idea that we could do something similar in Texas along the Central Flyway. So what our project seeks to do here is serve as a sort of first step. Obviously, if we want to have these programs in place, we have to have that data, the analytics, the beta testing to inform the development of that program. So we want to see if we can shed light on the path to a similar program here in Texas, as we are part of an important migratory flyway as well. So where we sort of started was, choosing an area of interest and learning about the different ongoing monitoring efforts that are being carried out by Coastal Bend Basin Estuaries Program. So what we have right now is 10 properties in Southeast Texas that have around 40 wetland areas that are actively managed in this way. So sort of strategically inundated at certain times of the year, such that they can support these migrating birds. So some of the data that CBEP has, our community partner has, includes bird counts at the species level. They've got metadata like characteristics about the habitats, moisture and water levels, etc. And so we're sort of using the areas that they're actively monitoring to choose our area of interest for the scope of this project. And what we intend to do is use imagery from these areas that are already being monitored to inform the model that we eventually hope to build. Because since these sites are being monitored, we know that they're doing their job. We know that these wetlands are supporting birds in the area. So we want to familiarize ourselves with those areas and try to understand what characteristics do they have, such that we could expand that out and identify other potential areas of interest for an eventual program, such as the bird returns program. So although we're not going to be working directly with the data here yet, or at least you're not going to see that monitoring data in the presentation, we do know that our community partner has a sort of systematic methodology that they're using when they go out and do this monitoring. So they're taking bird counts at discrete buffers. They're taking counts on a species level, so we have insight to the species diversity in an area. And what's exciting about this is that if we can eventually get this image segmentation model to work that we're going to look at today, then we might be able to correlate that with the bird count data in the future to not only identify the areas that have potential for wetland habitat, but also get an insight to how much potential they have. So if we know that there's a certain density of bird species and number of individuals in a given area, then maybe we can expand those numbers to the areas that we identify. So we might be able to understand how many birds could be supported, and that might strengthen the incentives that we would offer, to potential private landowners. So taking a look at our workflow for the project and getting an idea of where we're at as of now, we started with some data preparation and data pre-processing, which we knew would take a while, even just identifying the area of interest and learning about the different types of imagery that's freely available and their characteristics. And of course, we eventually want to go on to the model training and get our results. But right now we're sort of in the middle of these two steps here. So what we're going to see is that we've done a lot with the data preparation and processing. And if we felt confident in the data, we'd essentially be ready to press go. So we sort of have an idea of the type of model that we're going to use, a semantic segmentation model for classifying different characteristics such as land cover classifications. But the crux of our presentation is sort of going to be the roadblocks that we're experiencing now and why we haven't pressed go yet. So we're going to sort of dive into that today. starting with the satellite imagery that is available to us and what we've ended up working with so far. So in terms of freely available satellite imagery, that might be collected by a government agency, made available to the public, we have things like Landsat, Sentinel data, and the data that we're going to use is the National Agriculture Imagery Program data. So let's compare these different data sets. First, we have the Landsat 9 data, and the Landsat program has been going since the 70s. So we have lots of temporal resolution with passovers every day. The only problem here is that the resolution isn't so great. And we're going to see why that's especially problematic for our objectives. Because if we look at this image of Ward Island here, we can make it out. We're familiar with what this looks like. But that pixel size is so large with the 30-meter ground sampling distance that you really lose a lot of the features that would be characteristic for our purposes. The Sentinel-2 data is a little better, coming in at a 10-meter resolution of ground sample distance. However, when we look at the type of wetlands that we're talking about, which are inland wetlands, sort of managed and flooded for the purpose of just being a transient sort of wetland, you're not going to have the same characteristics that you would have of a coastal wetland system. So in a coastal wetland system, obviously we're looking for water boundaries, and you can obviously see those here. In a coastal system, you might have characteristic vegetation that you could use to make some classifications. But when we get into those agricultural areas, we're really lacking that. So we really need that fine spatial resolution. So that brings us to the imagery that we will be using, which is from the National Agricultural Imagery Program. And it's collected at a ground sampling distance of 1 meter. So we have this really high spatial resolution. And you can see that here in this image of Ward Island, that you start to see the coloration just in the water, right? You can see the different sediments that are suspended, seagrass beds, etc. And that would be really important for our purposes. So we're really happy with the spatial resolution that is enabled by these digital sensors that are considered high resolution with this extra near infrared band. And this actually isn't satellite imagery at all. It's collected by low-flying aircraft that are equipped with these digital sensors. And they do multiple passovers during the US agricultural growing season. So it's really crisp imagery. And what we're going to see is that we run into issues with the temporal resolution. So this is typically collected during the agricultural growing season in the US from May to September. And the data that the NAIP provides is already pre-processed. So what they've done is collected all of the data from those 5 months and done the pre-processing, done the filtering to remove anomalies such as cloud cover, and then they sort of mosaic all of those pre-processed image, and that's what is available to us. So talking a little bit about our study site and why we chose this area, and then we'll sort of go further into the methods that we use to extract the data. Again, we're basing our project around where the Coastal Bend Basin Estuaries Program is actively doing these monitoring efforts. And we have 2 sort of main sites that you could see are well separated. So I noted that there was about 40 properties that are actively being monitored. 10 properties that are being monitored, about 40 wetland sites. But we can sort of group those up here and here. Okay, so one near the coast and then one a little more inland in southwest of Houston. And so this is where our area of interest is going to be. Of course, we just wanted to keep the scope kind of small for this experimental project here. So we're going to be focusing on this area here. Then of course, we want to acquire the data. So we used the Google Earth Engine platform where the NAIP data is hosted. And to make that initial download, we just used the web-based code editor in Google Earth Engine. And we have some code here. Basically, what we have to do is indicate our dates of interest, which was the years of 2017 and 2018. And then we can plot some vertices to sort of put a rectangle around that area of interest. And what we end up with is this really large file that has all the imagery from our defined region. So this GeoTIFF file that we extracted using the code here is about 2.6 gigabytes, really large file. And it's just one file. It's not going to be amenable to our model. So we have some further processing of this initial extraction. But this code here just yields that area of interest. And I won't talk about it too much here because Justin's going to explain it a bit, but ultimately we end up splitting this into about 14,000 tiles. So Justin's going to talk about how we chose the dimensions of our tiles in the code that we used to do that sort of chopping here. Once all of the files were sort of extracted and processed in that way, we have them stored locally and in cloud-based repositories so that we could do the model when we're ready to get to that step. So Justin has some pre-recorded slides. I'm going to go ahead and play them. I think one of them might have been a little quiet, so we'll experiment here and see how well this goes. Okay, hold on. Let me turn it up a little bit for the folks in the room. Oh. Oh.
Now that we have our data downloaded locally, we needed a tool to visualize our GeoTIFFs, to explore the data, and make some calculations down the line. We decided to use Python, utilizing the Rasterio, numpy, and matplot libraries, which essentially graph our raster files while maintaining the spatial integrity of our data. As we move on to exploring and preparing the data for our models, we have found it helpful to plot the images as either the pixel number or as more recognizable units like length in meters as our axes. Additionally, we have also have the ability to modulate the colors, the RGB colors, to help kind of spot patterns easier, and have the ability to stack layers on the graph as well. With our new visualization tool, our first step was to explore the data further and take a closer look at the properties that our community partner actively monitors. Here, I have a small subsection of our data focused into one area with the hopes to visually spot some patterns that our model would look for. We wanted to see if we can visually distinguish the study areas, which we know are managed wetlands, from the surrounding fields, which may not harbor as many birds. We were really happy looking at these images. If we look at the top left, we see that the orange squares show the managed wetlands, and when we remove that layer, we can confidently guess that these are transient wetlands. We see evidence of previous water based on the types of soil coverage and the color of the plants in that area compared to areas that might be more dry. However, when we took a closer look at some of the other study locations, we found distinguishing these transient wetlands to vary in difficulty. On the left, we have 13 study areas highlighted in that orange. And when we took a look at the raw file on the right, we find that some of these areas had no distinguishing features, at least upon initial visual inspection, from surrounding areas which might not be productive transient wetland environments. And so, for example, if we look towards the top right of our images, we see that we have this large kind of collection of tan or beige colored fields. And our study areas within those tan or beige color fields have no distinguishing characteristics that look like wetlands. And so this was definitely causing us some worry and required us to look into this problem a little bit further. With some of these difficult sites to visually spot patterns, we decided to see what is possible by looking at some other sources of data taken at different times of the year. We wanted to explore the importance of the temporal aspect of our imagery, as different times of the year likely lead to different land use and plant cover. We decided to look at some imagery gathered in the spring, pulled from sources like Google or Apple Maps, and instantly saw a different scenario than what we initially saw in our NAIP imagery. Areas that previously looked just like barren land had clear signs of wetland activity, which showed that not only is image resolution a critical component in picking our dataset, but also the temporal coverage of our data. However, at this moment, we only have access to the NAIP imagery, as cost limitations prevent us from accessing other sources of high-resolution data. Now that we've explored the data a bit and highlighted some potential problems, we needed to prepare the data in a form that could be fed into our AI models. After looking at a variety of other land cover classification model examples, we saw that most models had limitations to the sizes of the images being used. Seeing this, we decided that our initial large GeoTIFF needed to be chopped into these small manageable images called tiles. We did so in Python using Rasterio, which we fed our initial GeoTIFF and cut our large image into small 256 by 256 square pixel images, which ended up producing more than 14,000 tiles. We utilized this size specifically as was actually the maximum size per tile our prospective models could handle, but we felt like it needed to be this large due to having adequate spatial coverage. Now that we have our data prepared into these manageable tiles, we needed to start annotating our images to provide the training dataset and to build the verification dataset for our model. We wanted to create a model that can classify each pixel as a different type of land, so we use this LabelStudio software, an open source labeling tool for a variety of AI applications and file types. Within LabelStudio, we utilize their manual labeling tool to highlight our tentative categories by drawing over the images that we fed into the software. We created multiple masks, one category at a time, that can then be combined to create a fully annotated image at the end of the annotation step. We expect this to be a very tedious step in our workflow, so we'll investigate the use of predictive machine learning models to assist in the classification with human guidance.
Okay, so thank you, Justin. Some questions and problems and challenges that are still facing us as we move forward into the fall. Obviously, the sort of focus of our presentation has been this issue with the temporal resolution. So that's our biggest outstanding question here. First, will it be enough? Okay, so what we of course want to do is follow through with the model with the image we have already. So we have all of this imagery collected. The annotations that you see were done very recently. So we haven't gotten through that process of, going through and annotating all of them. And we still want to explore if there's a predictive annotation tool that we can use. So of course, we'll try our model and we'll try our experiment as it stands with the imagery that we do have. But if that is not enough, then can we obtain some higher resolution data in terms of the temporal resolution? We feel pretty confident that 1 meter is good enough in terms of spatial resolution, as we can see in some of the images that we picked out to demonstrate that we can pick out some of those features with that 1 meter resolution. However, we're really going to need that temporal resolution, right? When we're looking at this NAIP data, it is all mosaicked over the course of the whole growing season, and that's going to be really problematic because when the image was captured for that region, was there water there or not? If it's been dry for a while, then you start to lose all of those characteristic features that we're looking for. And another question, if we do end up getting this data, is where do we go from there? Is if we're ultimately limited by the data availability, what future directions do exist for our project? And if we do get that higher temporal resolution data, then what will be the procedure for capturing all of this variation, right? Because does that mean that we sort of run the model for every day throughout where we have the imagery so that we can see when there was inundation and when there was not? That's something that we don't know yet. So that's an additional question that is outstanding as we move forward. And another challenge that we will face is this annotation phase. Okay, as Justin mentioned, it can be really tedious. We might be able to use some sort of predictive tool that we haven't explored yet. But we also see that the features that we're looking for are really fine, right? So there's going to need to be a lot of human intervention to make sure that we're not misclassifying things and that we're capturing all of the variation that we're looking for. So some of our fall semester project plans will be to run the model as it stands with the imagery that we have and see what it gives us, see if it's adequate. What we expect is that it will largely underestimate the wetland availability. So what we want to do then is try to connect with one of these commercial providers of satellite imagery, such as Planet Labs. We have seen that there are instances that students can be provided data through a student account with Planet Labs. It wasn't accessible over the summer, so what we're going to try to do is connect with this commercial provider, see about setting up a student account, and see what that does for us. Okay, and it opens up more questions if we actually get that data, because then we have to go back and revisit, right? If you remember the workflow, we already went through the data pre-processing and collection. We would essentially have to start back over, right? But at least we have the tools, we have some code written, and a much better understanding of how to do that data extraction, storage, and etc. And we don't know if more data will solve all of our problems, right? We might still be missing knowledge in terms of how these wetlands are managed, what it takes to have an area inundated at a certain time, you know, natural factors such as natural precipitation, natural inundation. There's still a lot that we feel we might be missing when we have, you know, the pieces starting to come together. So we're continuing to learn, continuing to flesh out our objectives, and continuing our correspondence with our community partner and our TAMU-CC faculty who have been advising us along the way. And as always, you know, you want to have a plan B, plan C. So we have talked about future directions if the image segmentation model isn't a go. Okay, if we're ultimately limited by the data availability and we feel like we're not getting at the objectives that we have, then we'll establish others. The bird program at Coastal Bend Basin Estuaries program has all sorts of different objectives, and you know they've been really great. I'm happy to see David Newstead in the chat. They've been awesome throughout the semester, so we're glad to be working with them either way. So I want to thank you all for your time, and I will open the floor up to any questions. Yeah.
Yeah, sorry, I always have questions. So NAIP data is 1 meter. It's really nice when you have RGB. And when I look at the image, the first thing really popping in my mind is, okay, for the farmlands, you may have sharp changes whenever there is harvest. So I don't know if you can just comparing for different seasons and just, you know, that's farmland that is of your interest, just remove those immediately. Only look at other interest regions. That will zoom in your focus a little bit better. That can be easily done with coding.
Okay. So you're saying sort of remove all the features that are obviously cropland and only cropland. Okay.
Yeah. Then you just focus on the possibility regions. That will probably save you some effort. Try to do the manual labeling.
Okay. So would that be like, so I have the 14,000 tiles. Would that be training it in a way that says, hey, remove everything that looks like this? Like remove anything that looks like just cropland?
So for example, what I'm thinking is your target is really looking at how wetland is changing, right? Your farmland will never becoming a wetland from 1 year to another because farmers keep their land, keep crops all the time. So if you can just look through the images from Centennial or something, and for multiple years, just look at how do you see the sharp changes over those regions, then probably you can just first initial process, remove those regions, and you don't consider those. Then you look at other regions because wetland is people don't touch, they change gradually. So if you see those lands have changing gradually, then okay, those are your potential target. At least that will remove large area of farmland from your images, and then start from there.
Okay, yeah.
And then I think that might save you some time.
Cool. Yeah, we could use all of the time that we could save. So I appreciate that. We'll look into that for
Because
sure.
in Texas, most of the land are farmlands, that area.
Yeah.
So
Yeah, especially if it was eventually to be carried out by someone else who looked at a larger portion of Texas. I mean, obviously we took such a small piece. So if that was an aspect of the model or the process, then it could, you know, increase the throughput, I'm imagining, of something like this. Yes.
Do you have any like confirmed images of transient wetland that you can use?
So yeah, so that's why we use why we centered our focal area around the monitoring sites, because we know that these are wetlands. And that they're deliberately managed in this way. So our community partner has agreements with these private landowners to come and do the monitoring because they know that the wetlands are there. So having this positive ID that they're in the field monitoring that site, then we can look at the same spot, you know, with different types of imagery. Of course, when you go on Google Earth, you are really looking at that really high res imagery. I think they use like MODIS and stuff. So you can zoom in and you can start to see some of these features. It's just a matter of if we can get data that you could, you know, have raw files for and put into a model. So there are characteristics that that we could point out.
Cause I was thinking like, if you had pictures of it when it was full-fledged wetland, you can find that time, you could run that first and then you'll have narrowed it down to see which ones are transient.
Yeah, so most of these from my understanding are transient in the sense that like they'll basically have like irrigation that they pull up and then flood, and then they get those sort of characteristics. But you can also have the natural inundation too. So yeah, we definitely just have to start with the confirmed sightings, and then, you know, maybe there could be some sort of negative input where you say like the croplands, for example, this is never going to be as
Could I ask a question?
yes, please.
Oh, hi. Sorry, just to kind of get back to the types of lands that we're talking about here. The vast majority of these of the habitats of interest are working lands. That means they are cropland, or they may be used as cropland in the growing season. So you wouldn't want to clip those out. That's probably the primary layer that is of interest here. But a good example of like the temporal issue is how rice is farmed, right? So rice is growing throughout the growing season. It's not habitat, and then they take their first harvest in usually around now, August, late July, August, and they'll do one of two things. They'll either reflood it and then get a second crop later, and in which case there's not really habitat for shorebirds until after November, or they may take one harvest and then cut it, and at that point they could flood it, and that flooded rice would be actually really good habitat. So the decisions that these landowners are making with regard to their harvest on croplands is one of the main determinants of whether there will be shorebird habitat or not.
Thank you for that clarification. I had a similar thought, like I just didn't really know how to express it as eloquently. But yeah, so that adds a layer of complication to things. But
the other thing, just while I'm on rice, is that rice is usually grown not always in subsequent years in the same wetland unit, or in the same, it's the same field, right, or the same rice unit to them, but in the off year it's usually fallow, and there are a lot of opportunities for turning that into shorebird habitat on the fallow year. So
yes.
What does the incentive look like for the landowners? Because at least what I think of, especially just like knowing people with agriculture and everything, to me, it sounds like, you know, if you put up your land for this, you'll have a bunch of bird waste in it, and then you still want to grow crops. And I can just see that being like a future problem in terms of fertilizers. And then like, I don't know a lot about birds, to be honest, but like, what if they like eat the nearby crops as well? And that can cause problems for people near, like the neighboring people?
Yeah, I haven't looked too much in like what incentives they actually offer and then what sort of promises they make to the landowners in terms of like how they'll ensure protection for their property, right?
Yeah.
So, I'm not sure how significant of a source the bird waste would be in terms of messing with the nutrient composition of the soils. That would definitely be something to look into. And, them being shorebirds, I wouldn't assume that they're like, and I could be just totally exposing myself as a chemist and not a biologist here. But I'm not sure that they would be foraging on the crops, but rather when there's the inundation, it's attracting these small invertebrates that are living in the flooded water, and that's what the shorebirds are eating. In terms of the incentives, like I think there are actual just cash incentives to say, hey, if you can hold off on growing this year, we'll give you a stipend so that, your land can be used in this way because it's really important. What I don't have any idea about is how strong of an incentive that has to be. What does the landowner lose if they sacrifice a crop versus a year? What is the financial loss there versus the financial incentive that is offered by the program? That would, all be future work to look into. But, it would be worth us looking more into the bird returns program since they have a lot of this fleshed out. So definitely by the end of the semester, when we start shaping this up and we start thinking about, imagining what this could actually be, I'll try to address some of those questions. So thank you so much. Yes.
Yeah, talking about, thank you for clarifying that habitat of the shorebird, but this brings me another thoughts on how to use the Sentinel-2 data, because there are papers regarding how to use that data from multiple channels to retrieve the soil moisture. So if you can get the soil moisture data, if retrievals, and then you look at very high soil moisture, maybe that's the water surface you can identify by using Sentinel-2. And I didn't really look in detail, but maybe that worthwhile to do a little bit of literature reading and see if that is a possibility.
Yeah, that would be great, especially because, the Sentinel is going to have the much higher temporal resolution, which would be awesome for, if we really are just looking at imagery features that are a result of inundation, then we could, infer that from the moisture level data. So that's a really good thing for us to look at. Do you think we would still add that with the imagery data, or would you suggest that as a different route for this?
Well, the retrievals are also image data. It comes out to 2D array. So it's just what you need to find is if you can use that to find all the inundation area, and then maybe that can be related to a topic.
Cool. Thanks so much.
I'm just gonna say something that may be completely irrelevant. Remy had the idea that you can look at like ag exempt, like obviously people file for ag exemption. There are a lot of subcategories under that ag exemption, but that data should be somewhat publicly available. I'm not sure what it looks like. So I'm not sure if it just says, oh, ag exempt, timber exempt, and that's it. Or if it says ag exempt under farm animals or ag exempt under crops and like what kind of crops. Because you have to explain your exemption when you're applying, obviously. Probably most of this land is under ag exemption, so you may be able to figure out what ag exemption is associated with these like locations or coordinates. And then that could give you an idea of what might be potential, like, if they are ag exempt under growing like things like rice or something like that, then they're potential, people that would be interested in this kind of program. And so their land might be more interesting than someone who's exempt under timber, who is probably not flooding their land, right? So that would maybe like help you narrow it down as far as like the types of cropland that are in this area. But that may be like a rabbit hole you don't want to go down, because it could be a totally absurd data set.
It would definitely be worth looking at. That's a good point, just seeing how the different land types come into play, and if there's some sort of trend in the monitoring sites, and what exemptions they have or how they typically use their land, that could be a good step for us to take.
So obviously ag exemption itself is an incentive, because that's like a tax, whatever, write-off or whatever. So if it's available for them, they're probably doing it, and so then they're telling you something about their land.
Right.
But again, seasonally, they do different things with it, so it's like, the exemption could be one for one thing, and then they do something else too, so. It's not necessarily like perfect, but it could give you some, it could rule out some things that are maybe not ag exempt and like actually not cropland, and just like someplace that an oil company has like a
Right.
small like caliche patch, you're like, okay,
Right.
well, that's never gonna be of interest to us.
Uh-huh.
So then you could rule it out.
Cool. Yeah, no, that's a great suggestion. Thank you for that. I'll have to look into that a bit, see what's even out there, you know?
Yeah, I'm not sure, so.
Yes.
On the flip side, oftentimes when farmers use crop rotations or, have an off season for their land, for the soil to recuperate from the harshness of having one single crop, often that actually increases benefits to the next crop, makes the nutrients of the soil more rich, and usually the crops are more disease resistant as a result of all the excess nutrients. And I was wondering if you knew if that was also the case for wetlands and rice. And this is like not just a question to you, but also to your stakeholder, if you know as well.
Yeah, I'm not too sure about some of those dynamics and would come into play here. Maybe David has a comment on that, but yeah, I think just based on how the area is used at different times of the year, there can be different benefits, either in terms of growing crops or for the wetlands. If you end up with a really good nutrient profile, from the last growing season, and then you have the flooding come in, and then you have high nutrient availability, maybe that creates the perfect system, instead of just having, flooded waters that are maybe, not supporting the primary production. So that could be an important aspect.
Yeah, there are incentives through NRCS, through the Farm Bill programs, a number of management practices that landowners can do and get reimbursed for, or be paid an incentive for, that have to do with soil health, like you mentioned, cover crops and things like that, rotational grazing. There's a number of different things, but there's also there's some literature that shows downstream water quality benefits from leaving fields fallow, obviously, and for having shallow wetland habitat temporally. But so there is a practice called shallow water habitat management, but the temporal the landowners can basically get this from providing that habitat during the midwinter months, which is when waterfowl are around, and that's also an economic driver for some of these farmers. The problem with it is that the deficit in shorebird habitat is that shorebirds migrate earlier, they migrate through, and then they migrate later back in the spring. So benefits that accrue to waterfowl do not necessarily translate to shorebird benefits. So that's where the temporal aspect is really important. And that's something that the incentive programs that are currently available miss.
I just had a thought about the incentive programs, like we were saying. Is there a restriction on what kind of pesticides they can use on the fields they intend to flood? Because I can imagine if you like covered pesticides and flooded, that probably wouldn't be the best.
Yeah, I don't know. I imagine that's something that like you would consider in a formal program like this, is what is, are we gonna try to enable habitat that isn't actually safe for the birds? So I imagine that could be something. Again, it might require more incentive to convince the landowners to, hold off on using certain practices, just based on how they've typically operated. If this is someone who's stepping into this for the first time, they might have some resistance to the applications that they're going to use in their normal operations. So it could definitely be something to look into when the program, is developed, is how do you manage all of those different considerations, for sure.
This is another really big question in the shorebird world, the effects of pesticides, and it's really difficult to trace through migratory birds. You can poison animals in a lab setting or something, but these wild birds that are constantly moving through, it's very difficult to trace the any potential contaminant effect. But it's really there are a lot of people scratching their heads and trying to figure out how to answer this question. But yeah, ideally, you would assume that there probably is some negative impact from heavy pesticide use or certain pesticides, but until we can quantify it, it's really difficult to get at. But eventually, if it's significant, then there's things beyond incentive programs for producers, like certifications for, say, bird-friendly rice or something like that. There's examples of bird-friendly coffee, bird-friendly beef, things like that, that ideally would provide the producer a market benefit in the market as well for their product. But we're nowhere near that yet.
Yeah, okay, last thought, this is also not a question you might be able to answer, but how might like a drought season affect sort of this kind of incentive program? Like if there's a drought, they're already having trouble watering their crops or whatever, like I'm not sure, and I'm sure they use well water to flood the fields or something like that. They may not be super interested in doing that if they're like running the risk of their well kind of like running dry. Now that's been like a problem in the hill country, like recently, like the drought's been decently bad. Lake levels are super low. I'm just not sure if this particular area is affected, but some areas of Texas obviously are fairly dry most of the time, but still in that flyway. So like during a drought, the birds are gonna have trouble anyway. And you might also have people being like, I can't do it this year. Like,
Right.
So I'm not sure.
Yeah, that would definitely be something to navigate. I mean, if from all directions, you're being told that you can't use this water, because you don't have it. Maybe there's some regulations to say, hey, we're in a drought, you can't be doing this. That would definitely be something to navigate even on just like a seasonal basis, right? It's just making those assessments and seeing, do we have to sit out this year? Do we have to try to shorten things? What would the dynamics of that be? That would really come down to the development of the formal program, right?
It's like considering your photos, like I don't know what year they're taken, like if that year happened to be a drier year, and so you might have trouble seeing those characteristics because it was a drier year. So the
Right.
natural inundation isn't gonna be as prominent even.
Uh-huh.
And so you might like, if your photo is from, last year, you're like, oh, this is a lot more difficult than it was 20 years ago where we like have the like really clear
Right.
water present.
Yeah, it could even just be interesting, like if we think, I don't know, 20 years from now, if something like this were to get developed, then that might be something that the high resolution informs, on a sort of active basis of what are the consequences of the drought? What is the most that we can do while dealing with this situation that just happened this season versus the next? So I think if something were to be formally established, there would, that would have to be something to take into consideration for sure.
The way the bird returns incentive program and other ones that are similar in objective is that it's like a reverse bid auction process. There's a certain amount of money that's available, and landowners will put in bids and say they can provide this much acreage at this time, and this is how much it's gonna cost. That's my own costs plus some fee for kind of inconvenience, and those bids are all pooled and then selected based on what makes the most sense for the most habitat. So, and you would assume that in a drought year, there would be a higher priority. Your habitat would fetch a higher dollar, just to put it in kind of market term.
Anything else from anyone? Okay.
Great job, Justin.
Thanks so much. Thank you much for all of your insight. It's really helpful, and congrats to my peers on the summer term. 5 p.m. today.
Thank you, everybody, for joining us, both in person and online. I do apologize that the recorded audio on the PowerPoint slides couldn't be heard by those on Zoom. We will be posting the recording of the presentations in our stages at TAMU-CC YouTube channel. And I will see if I can get the audio into that. If not, I have emailed it to the PowerPoint to everybody with that with the audio recording. If you have any additional follow-up questions, please feel free to email our students directly, or you can email me and I will forward it on. But again, thank you for joining us and have a good day.
Mhm.